Good morning. Much better. Okay. Good morning. Welcome to this conference. One important problem in infectology is to understand how viruses are able to evade host immune defenses. Mariano García Blanco is a professor in biochemistry and molecular biology, which research has been focused on RNA splicing and in the study of RNA-protein interactions, how they regulate gene expression in cells and viruses, mainly flavivirus, dengue, yellow fever, and Zika viruses. His group has identified human and mosquito RNA-binding proteins that impact in flavivirus replication and help to understand how these viruses evade human and mosquito defenses. Mariano García Blanco obtained the AV degree in biochemistry at Harvard College, is MD in Yale University, PhD in biochemistry at Yale University, PhD in molecular biology at the MIT in Cambridge, and among other positions, he has been director of Duke Center for RNA Biology, and actually he is professor and chair of biochemistry and molecular biology at the University of Texas Medical Branch. He has published his work in the most relevant journals such as Nature, Cell, Proceedings of the National Academy of Science, Journal of Virology, Genes and Development, etc., etc., etc. Today we have the pleasure to present him, and he will talk about a summary of his work on host protein RNA interactions under the title that is in my back, The Love-Hate Relationship Between Host Proteins and Viral RNAs. I would like to thank Professor Mariano García Blanco to be here, and I hope that his stay in Salamanca will be helpful and joyful.
Gracias, Rodrigo. I think I'm wired. Primero, voy a decir unas pocas palabras en mi versión del castellano. Muchas gracias a todos. Muchas gracias por tener una ciudad preciosa y una universidad impresionante. Y ahora cambio al lenguaje, mi lenguaje adoptivo, el inglés. So now I'm switching to my adoptive language because I think it would be painful for you and for me if I actually gave this lecture in Spanish. So let me first start with a very important point, and that is to thank Antonio Muro and the other organizers for this great opportunity. So I'm going to spend 15 seconds or 20 seconds telling you that it means an enormous amount to me for many reasons. Yesterday, it meant a lot to me to introduce my mentor, Phil. It's a special moment to share with him. But it also means a lot to me because my father walked down these streets. He met Unamuno, and I read Unamuno's works, and Salamanca was like Rome for me when I was growing up. It was the place that you went to learn, and it was a center. I like it a lot more than Madrid. I'm sorry, Madrileños, but so it also means an enormous amount for me, and this is the last personal bit, because both my great-grandfather and my grandfather were pharmacists, and they started the first pharmacies in the Caribbean, in Cuba and Puerto Rico. So, unfortunately, I'm not a pharmacist, but I'm going to try to tell you what I'm going to tell you today from a point of view of the role of pharmacy in medicine. So the challenge that we face, those of us who are interested in emerging viral infections and re-emerging viral infections, and I'm going to mostly focus on acute infections, but there have been very good examples of infections that are more chronic, like HIV infection, which also have enormous epidemic potential. So how do we address these infections of high epidemic potential? So the way I'm going to divide the talk, and this is my guide to you for when you can go to sleep if you want, is I'm going to start by telling you about the viruses that we work with. But I'm going to actually suggest to you that we are using these viruses as models for other RNA viruses. And why RNA viruses? I think if you look at the last 150 years, some of the most severe, not the only, but some of the most severe epidemics that the world has seen are caused by RNA viruses. And we could spend a whole lecture talking about why that may be the case. Then I'm going to go into a more technical third of the talk. And if those of you who don't care about RNA and proteins can doze off, and I'll tell you that that third represents my personal obsession to the idea that RNA-protein interactions are critical both for the mechanics of the virus, but also for the way the host responds to the virus. And this is an obsession I share with many other scientists. Others think we're a little crazy, but I will talk to you a little bit about this mechanism that flaviviruses use to subvert the innate immune system and how important it is. And it has very important applications, for example, for vaccinology. And then I'm going to end with something that I haven't spoken outside of the United States and Singapore, and it is a joint initiative by a small group of scientists we're now expanding it, and we call it the Global Rapid Biodefense Response. And it essentially hopes that we can deal with future epidemics in a more rational and proactive way than just telling people, stay home, be quarantined, let's take your temperature when you go through the airport. And already many things have happened in that direction, but I think we need, these viruses are like doctors without borders. They don't recognize borders. These viruses go all over the world, as we saw recently in several cases. So we need to develop a global response. So let me start with the most important insect-transmitted virus in the world, and that's dengue virus. Dengue virus is actually caused by 4 viruses, not by 1. There are truly 4 different species of dengue viruses, which are known as serotypes, but they're really 4 different viruses. And they actually have subtly different biology, and very good clinicians can tell you that they have subtly different clinical presentations. Dengue 2 may be the most severe. It is also the most successful of the dengue viruses. It has spread throughout the world and infects the most number of people. What makes dengue a global threat, it's not the mortality. The mortality of dengue is actually low. It's below 1%. It is enhanced if you've had a previous infection with another dengue virus. But even then, the mortality is very low. But what makes dengue so amazingly important is that there are 400 million infections every year in the world with dengue viruses. Of those 400 million, of those 400 million, 100 million are ill, and the burden to society is quite large. They are transmitted by Aedes mosquitoes, Aedes aegypti, which is like our domesticated mosquito in most of the parts of the world where dengue is endemic. And actually, Aedes albopictus, which landed in the port of Houston back in the 60s and is now spread all over the eastern third of the United States. And these mosquitoes both are capable of transmitting dengue. All of the viruses that I will address directly today are transmitted by this mosquito genus. And the fact that this mosquito is domesticated, it really likes to live in cities, has changed the complexion of dengue. As globalization has made people move around, large cities, mega cities in Asia, dengue has found a perfect place. And that's why it is exploding in the last few decades. For you Europeans in Salamanca, you don't have to worry about Aedes yet. I don't know what's going to happen in the next 100 years, but for now, but you have a series of flaviviruses here that are transmitted by ticks, garrapatas in case you don't know, and those are very severe. So there's a whole family that I won't talk about. And there are other diseases transmitted by other species of mosquitoes within this family. So the second, and maybe an excellent example of how little we know, is Zika. Zika is named, actually misspelled, because it was found in a forest in Uganda that is Zika with 2 eyes, but I guess it would be difficult to pronounce. Zika virus, it's clear that it moved centuries ago and that it has been endemic in Africa. There's evidence that it was in Asia, but very little was studied about Zika. It was ignored until the explosive pandemic of 2015, 2016. And we're now, for example, 100%, some people claim that 100% of people in the Caribbean have seroconverted to Zika virus. Again, very much like dengue, maybe even more than dengue, there are many more asymptomatic cases than symptomatic cases, and that is a very important epidemiological concern. The thing that Zika taught us, and these are the images that you saw in the newspapers of the very, very unfortunate babies born with microcephaly or aborted spontaneously. It taught us 2 things about flaviviruses, human flaviviruses, that we had only seen in animal flaviviruses. Number 1, transplacental transmission, vertical transmission from the mother to the child. And number 2, linked to that is this propensity to infect the genitourinary system. The field has been obsessed with neurotropism, but the real tropism that's important in Zika is the genitourinary tropism. In fact, it is present in the testes of men for many weeks after infection, or it can be. It is found, actually live virus can be found. The mouse models and the primate models show exactly the same thing, and it can be sexually transmitted. So this genitourinary tropism of Zika is of great interest to us. And it's something that I won't have too much time to talk about, but it taught us about a new form of transmission. Even if you eliminated mosquitoes, Zika could still be passed from one human to the other. There is no evidence that this happens with dengue or yellow fever. And then my favorite of these viruses, because for a virologist, this is a real virus, because it actually is very lethal, is yellow fever. Yellow fever, essentially today, is only seen in Africa and South America. It is a hemorrhagic fever in which and we'll talk a little bit about host genetic variation, but about anywhere from 5 to 30% of individuals can die of yellow fever infection. 200,000 cases, this is all extremely approximate numbers because it happens in many places in the world where there's not good epidemiology. And about 30,000 individuals die every year. This happens, and I want to harp on this point, because this happens despite the fact that one of the best vaccines in the world, in fact, a Nobel Prize was given for the discovery by, I think it was Mike Thaler in the 1930s, came up with the vaccine that is essentially the same we use today. And it is an example of a super effective vaccine. It gives you lifelong immunity. The WHO finally saw the light and changed the standards. You only have to get a shot once. But even then, 30,000 people die. So we'll talk about that in the last part of the talk a little bit, about why that is and what can be done to help that. When I'm giving this talk in the United States, I actually like to remind Americans that we suffered from yellow fever, particularly in the southern part of the country, although it went all the way to Boston at times. And here is two Yankees, both from Connecticut, who came down south. One died in North Carolina, one died in Texas of yellow fever epidemics. And these epidemics were relatively frequent in the United States. So these are the 3 viruses that we use as our 3 viruses of study. So what are these viruses? With my apologies to those who work on lipids. So actually, before I go on, let me tell you one more thing. There is not a single approved specific antiviral agent for any of these viruses. So challenge for pharma, challenge for all of us is either making effective vaccines for each one of them or having effective antivirals that we can actually use in cases. particularly if you have someone who's sick with yellow fever. By that time, the vaccine is too late. What do? Right now, you can do nothing. You do supportive care. So what are they? They are RNA protein machines. They have a genome that looks like the messenger RNA of our histone genes. It is capped. It doesn't have a poly-A tail. And it has only one open reading frame. So there's only one protein synthesis unit that gets cleaved into 10 different proteins. And this cleavage and all of this very complex architecture is one of the biggest challenges this virus faces. And a project in the lab that I don't have any time to talk about is about how the virus uses special host proteins to be able to insert itself into the membrane and to cleave where it needs to cleave and to fold the way it needs to fold. These proteins produce the proteins, for example, this is a polymerase, the proteins that are necessary for replication. These are the structural proteins here on the left, is three that form the viral particle, and the others are all proteins involved in replication, and many of them also have roles to dampen the immune system of the cell. This is a very brief life cycle that can be, this is for dengue, but it's really very similar for many flaviviruses. One huge problem in terms of developing, for example, molecules that block binding to the receptor or therapeutic antibodies that block binding to the extracellular receptor is that the majority of these viruses use many different proteins to get into the cell. So if you block one, they come in through the other. So the viruses actually bind to one of many different receptors. They then go into endosomes, and upon acidification of the late endosome, and here there is a point of specificity which people are actually targeting, then they release their genome, which is also a messenger RNA, because this is a positive stranded virus. And that immediately then is recruited to the endoplasmic reticulum while it's translated into proteins. And everything happens in these compartments that the virus creates that are surrounded by endoplasmic reticulum membranes, and here you have both replication, and then in nearby compartments you have assembly. From an RNA-centric perspective, and this is just to introduce the second third of the talk, this viral genome is a very versatile RNA, right? The viral genome can translate, so it's a messenger RNA. It's what makes all the proteins of the virus. It uses it uses our translation machinery, but it has unique susceptibility to certain ribosomal proteins. So potentially an interesting target, but a difficult one. So the translation occurs almost 100% in association with the endoplasmic reticulum. The RNA can also serve as a template for RNA synthesis. So the virus goes from the positive strand, and using viral replication machinery and unknown host factors, it actually makes a negative strand, which then creates further progeny. The positive strands then come out, and they can be sorted into any of these activities. Eventually, positive strands get packaged to form new viruses. But there is one other, and actually in terms of mass, a very important pathway. Anywhere from 50 to 90% of these genomes actually get degraded. They get degraded by a mechanism that's not completely understood, because it involves a decapping reaction, which is not understood, and then a degradation reaction mediated by an exonuclease, XRN1. This exonuclease cleaves, so it seems incredibly wasteful. You get 90% of your genomes and you destroy them. And you destroy the genome, but the last little bit of the RNA remains intact. And this is a non-coding RNA. It's a non-coding RNA that, as many of you have heard, and this is one of the things I want to try to emphasize, is that we have to think of RNAs as targets. Proteins are not the only targets there. This sfRNA has a very interesting structure. Jeff Kieft in Colorado has determined the crystal structure, some of its parts. We're collaborating right now with Wat Chu at Stanford to get a whole view using cryo-EM of the whole RNA. This RNA then is made in great numbers, and that non-coding RNA is essentially an anti-immune RNA. Okay, so I want to summarize here because eventually there are many people interested in drugs for these viruses. And we can talk about the great challenges of using therapy in an acute infection. Nonetheless, the virus has all these different processes, mechanisms of viral replication, which use enzymes that we don't have. The RNA-dependent RNA polymerase is a target that has been tested. There's a viral protease that has been tested. We know from the hepatitis C literature which is our distant cousins of these viruses, that those could be targeted. Up to now, it has been a failure. But that doesn't mean that we shouldn't continue doing that. We also should be targeting host immune measures. I think yesterday was some discussion about this, that we know that many times the hemorrhagic phase of yellow fever and the hemorrhagic phase of dengue is as much a product of the virus as it is of the host. So it is very important that we don't forget that we may want to, early in the infection, enhance immunity, and then later in the infection, reduce it. So thinking of pharmaceutical formulations that actually change the therapy as we go. And several people, particularly at UNC, are developing this type of approach. And then there are many viral, we're going to focus, anti-immune countermeasures that if we actually could inhibit those, we could make viruses that could be maybe vaccine candidates, and we will talk about that. Or viruses that if we could do it during infection, we could inhibit the infection. Equally, some of the host immune reactions we may want to increase at the beginning, and several people are involved in the protein called RIG-I and getting RIG-I activators, small molecule activators of RIG-I that will enhance the immune reaction, at least at the beginning of the infection. So let me summarize very quickly this part. I told you about flaviviruses, flaviviral RNAs, and RNA binding proteins, both from the virus and the host. And this is a very RNA-centric view of this. And I won't read everything here, except that I will tell you that many of the mechanics and of the measures that deal with immunity in these viral infections are mediated by RNA-protein interactions. Charlie Rice at Rockefeller did a beautiful screen of proteins important for innate immunity, for actually the second layer after intrinsic immunity of defense to viruses. And he found that something like 50% of all the important gene products were RNA binding proteins. So we are going to use these viruses as models of acute infections. So now I'm going to get slightly bit more technical, but I'm going to summarize. Some of this has been published, so I'm not going to go into great detail, but I'm going to tell you about a series of interactions with that non-coding RNA that we call the sfRNA, and how we think those are really important for the virus. And I'll show you one specific example where it has been applied to making an attenuated vaccine. So let me give credit to the people in the laboratory or laboratories, because these individuals some of them were in Singapore, some of them were in Texas. Alex, Cattell, so they're all postdocs except for Cattell, who was a graduate student, and now Julian is my co-PI in Singapore. So these are the individuals that did a lot of the work I'm going to show you. So back to, this is work of others. This is just to show you that you actually get this degradation of the whole genome to make the sfRNA, and you actually make several different versions of this sfRNA. And interestingly enough, in mosquitoes and in humans, you make slightly different versions of the sfRNA. And essentially, the cleavage ends at several beautiful structures that are very, very stable and stop the exonuclease. So think of it as an active molecule. It's about depending on the flavivirus, about 400 nucleotides long. So it is a medium-sized non-coding RNA. I don't like that nomenclature, but, and it is, you know, for many years it was thought to be anti-immune, but nobody knew exactly how it worked. So what we did was we took this is a two-dimensional view that actually has some representations of the X-ray crystal structure that was determined for the equivalent. This is dengue 2, but this was the equivalent structure for Zika virus. We took this RNA and we said, what are the proteins that bind to it? And in particular, we were interested in the proteins that bind to these two structures, because these two structures are the most rapidly evolving structures and sequences in all flaviviruses. So very quickly, I want to tell you, we've discovered many different proteins and published on several of them, but I'm going to give you a few highlights. The first ones are 3 proteins that are absolutely required for an effective interferon response. So this was immediately important. This is the curves of interferon activity that you get when you have a control or when you have 2 independent sets of siRNAs knocking these proteins down. And essentially, interferon is much less active when these proteins are not there. This was done by Alex, Ward, and Cattell, and then Cattell went on to show, and I'm not going to belabor this in terms of the data, I'll tell you what the model was, that these proteins are critical for the translation, for the formation of all of those proteins that interferon induces. So when we looked at the cells that did not have these proteins, interferon still bound to the receptor, interferon still went through all of the signaling cascades, the interferon-induced genes were stimulated at the level of the mRNA, they went into the cytoplasm, and they were not translated. And the mechanism appears to be a mechanism of competition with other mRNAs. Cattell showed that if you actually introduced sFRNA exogenously, and also using mutants of the virus that either formed sFRNA that could bind or not bind those proteins, that this cycle was happening. If the virus makes the sFRNA early and robustly, you inhibit these proteins and you inhibit the interferon response. Actually explaining some very old phenomena in the field about how soon you had to add interferon for it to be active against dengue. So obviously, this is an interesting, if we could inhibit this reaction here, we would make the virus much less pathogenic. And so we bombastically said, okay, we have a strategy. This is going to happen. Every flavivirus is going to bind these 3 proteins. And then we found that that was not the case. And actually, it became even more interesting because, and I will not show you all the data we have for this, but here, these are the 3 proteins. And in a Western, here are dengue 3, doesn't bind them. Kunjin is a type of West Nile virus, doesn't bind them. Yellow fever, either the vaccine or the pathogenic doesn't bind them. So we modified. The data always trumps your best ideas, and we modified our strategy and said, okay, this may be a general strategy, but each virus will use different tactics. And I can tell you that we now have tested this, and I think we have proved it, that different viruses actually sample different anti-immune, pro-immune proteins in the host and inactivates them. So yellow fever doesn't care one bit about these 3 guys, whereas it cares about another protein that activates the interferon system in a different way. So, but perhaps the most important and interesting test of this hypothesis came from a collaboration with a terrific colleague in Singapore. He's a physician, an infectious disease specialist, an epidemiologist, and now a molecular biologist, virologist, I should say. So En Young and I went back to, and Young started this from an epidemiological perspective. He sequenced hundreds of strains of dengue virus from Puerto Rico. There had been an epidemic, complete coincidence. It's not because I was born there. It was the most complete collection of viruses that in the 1990s there had been a change between one type of dengue 2 and another one. This is not a serotype. This is not 2 different viruses. This is one genotype, so one subclade took over, and the subclade that came in, and we don't know exactly where it came in, but it's probably from Asia, that subclade was more pathogenic and much more epidemic. And it created a big outbreak. And Young looked for a while, and he looked for where there were changes in this virus, and he couldn't find any protein where there were changes that were actually correlated with this. But there were 3 changes in this sequence, which is a sequence that encodes for the sfRNA, for this non-coding RNA. And very quickly, we actually went on to show that, that this changes accounted for the increased pathogenesis of this virus, because it actually inhibited interferon by a different mechanism, and also enhanced its ability to grow in both human and actually mosquitoes. I won't show you all the data for mosquitoes, but this virus is actually has much higher transmittability. So we were able to, in the laboratory, recreate a credible explanation for enhanced epidemiological fitness in these viruses. And the model is very simple, and it only explains in part what happens, but this explains a large part of why these viruses are different. This non-coding RNA binds, the one from the epidemic strain binds a protein that's absolutely essential to maintain high levels of interferon and inhibits it. Whereas the one that was there before, the endemic one, doesn't really bind that protein very well. And therefore, there's a more robust interferon response. And this difference accounts for a large part of this epidemiology. And we published this both in Science in 2015 and later in PLOS Pathogens in 2017. So that was actually very, very exciting to us. But what about Zika? Everything I've told you is more or less on dengue, and obviously Zika came around, and all of us in the community who were working on flaviviruses decided that we had to work on Zika. So in Zika, the lessons we've learned, and I did say to someone that I was going to show an old-style northern here. We like to do the northerns to see both the genome and the subgenomic RNA, this non-coding RNA in the same gel. And essentially, without worrying about the details, we actually identified the proteins that bind Zika, and we're able to show that this protein called FMR1, or the product of FMR1, FMRP, is a very important binder. It actually is involved in intrinsic immunity of the virus, and interestingly enough, we actually explained in part why this virus, why if you muck around with that sequence, you make this deletion here, and the original deletion was made by my colleague at UTMB, Pei-Yong Shi, who's a terrific molecular virologist who we recruited from Novartis in Singapore. And we were able to show that part of the reason that this virus was attenuated was because it couldn't bind to FMRP. So if we could eliminate FMRP, the 2 viruses behave about the same. And this is scientifically very interesting. I was thrilled, so was Pei-Yong. But the importance is that this is going to be the lead attenuated vaccine for Zika right now. So it's already shown incredible efficacy. It's actually in humans right now. It's shown efficacy in primates. I have to give credit to the old group at NIH that did something very similar with dengue. So this is, Pei-Yong essentially. So I will come back to this idea that we now have learned a general way of making vaccines for flaviviruses. So if a new one comes tomorrow, we may be able to make a vaccine very, very quickly. So this is the model. Clearly, FMR inhibits translation of either the virus or proteins necessary for the virus, attenuates the virus. We've done this both in cells and in animal model systems. I don't like the dengue and Zika model systems in rodents, but everybody uses them. The only model I believe is the testicular infection, because it's exactly what you see in humans. And there we could see that if we attenuated the virus, or if we looked at the targets of this protein in the testes of infected mice, they actually were consistent. And this is, hopefully will be coming out in the next few weeks in eLife. So this is the general hypothesis, and this is the end of this more technical third of the talk. What we believe is that this variable region in the genome is there to inhibit the innate immune system particularly, or to inhibit intrinsic factors, and that every virus or even every genome, every genotype will actually affect different components of the immune system, dampen it, and then together with other mechanisms of the virus, make the virus grow better. Obviously, we have to actually consider that there are other ways of, no implications here on transmission here. This was just for enjoyment, but because I actually usually have Donald Trump here, and then I have the Prime Minister of Singapore, but I decided that enough press is not a good idea. But what I just want to mention is everything I've told you today in humans happens in mosquitoes, except obviously they have a very different innate immune system. So the perplexing finding that we have is that this non-coding RNA, you can find it in mosquitoes, you can look at it in northerns, it looks a little different, it's a little different size, but the critical finding, and it's not important here what these two viruses are because we find pretty much the same finding for both, except at a different level, as expected, because this is a more epidemically fit virus than this one. But the dramatic thing is that compared to any other tissue in the mosquito, in the salivary glands, there is almost 100 times more of this non-coding RNA relative to the genomes. So we were perplexed by this and suggested that maybe salivary glands are obviously important because that's the organ that eventually produces the saliva that gets the next host infected. But it occurred to us that there's another possibility, which was intriguing, and we believe it's actually correct, which is that the sfRNA actually in some form or another is actually secreted into the saliva. And that actually that goes in together with the virus and sets the terrain of the infected cells. The only way that this makes sense in terms of molarity and numbers of molecules is during mosquito infection. Because at that time, if you've ever seen a movie of a mosquito biting, it bites a very, very small area. Once it expectorates in a very small area. And the number of cells that are infected are very few, probably are infected at a relatively high MOI, and you want that infection to be as robust as possible. And so we think we calculated you can, if we're right, and this is some of the data that we have up to now, that we can actually find this non-coding RNA, both by RT-PCR and also by sequencing in saliva. Don't ask me how we do these experiments because it takes hundreds of mosquitoes to be plugged into a little tube where they expectorate into media, and then you collect those and pool them. But here is essentially in salivary glands, and this is what we actually see in the actual saliva of the mosquito. We know that this sfRNA in saliva is resistant to micrococcal nuclease unless we treat it with detergent. So we're now purifying the vesicles, whatever it's in, but we are predicting, because salivary glands are known to make large numbers of exosomes, that it is within exosomes. So we believe that the mosquito is actually injecting this into the host, which again extends the hypothesis and maybe would extend things we can do about it. Okay, so this is why I'm so obsessed and why I think these RNA-protein interactions are so important for these viruses. I could argue the same for many other RNA viral families. But now so now it's a pitch, particularly for those here who could influence policy, who could influence the Spanish government or their government. Many of you from Latin America share these problems, so with many parts of the world. So my colleague En Young Oy and I decided that we were tired of hearing about, we have a lot of epidemiological colleagues, and we were tired of the old type of what we call reactive epidemiology. And we're not alone in this. We just, there was a very nice meeting of the Chinese Academy of Sciences and the National Academy of Medicine in the US that essentially, where I made this proposal already. But it's essentially, how do we know what we don't know? In other words, if you were a Jesuit priest in Arizona, in the United States, the day before a smallpox epidemic started, how were you going to know that, right? What were you going to do? And the Jesuits actually marked every death, so these are beautiful curves. And then was, shocked. Right? So what we're trying to do is very hard, which is to try to be ready for something that we don't know what it is. We'll see. So we've roped 3 institutions already. The next big institution we're trying to rope is the Wellcome Trust, because we have both Young and I happen to know the director very well, and he's really interested in this sort of stuff. And I'm going to give you a few examples. The first steps is RNA viruses. I've already alluded. They're responsible for many great epidemics in the last 100 years. We would like to have evidence-based epidemiology that considers both variations in the host and variations in the pathogen. We would like to have generic vaccines for classes. All of this is relatively obvious. Many people have talked about these things. We're just trying to implement it. And I think we also have some inside information about some of these things in the legal way of inside information. Some of our work has led us to this. Therapeutics, and I think here we have to consider small molecules, biologics, both monoclonal antibodies, but also nucleic acid-based biologics. And then for transmitted agents, because remember now this is going to be general for RNA viruses, biological countermeasures. For example, a beautiful work of Scott O'Neill in Australia, that if you infect mosquitoes with Wolbachia and release them, they've really now shown in massive epidemiological studies that you can decrease transmission of some of these diseases. The question is it going to be sustainable? And then for zoonoses, we absolutely need a One Health approach. So it's not only medicine and pharmacy, it's veterinary medicine too. So very quickly, just examples, some from our work. Why considerations of the host? This is an epidemic of yellow fever in Memphis, and this is the mortalities. The populations between Caucasians and non-Caucasians were about exactly the same in the city at the time. This is the mortality for Caucasians. This is the mortality for non-Caucasians. The case fatality rate was 7-fold higher in Caucasians. And now we're trying to explore, we think we know the pathway that was different between the two, because there are important genetic variations between Caucasians and non-Caucasians in innate immunity. If you measured cholera, it was the opposite. Okay. So we have to have, and here's what, back to some of the things Phil mentioned in his talk and yesterday to the press, the more we understand about our genetic makeup, we may be able to tell which types of individuals are going to be most severely affected by something. These are socially difficult questions, but from a scientific point of view, we have to answer them. Generic vaccines. We think we know now how to make a generic flaviviral vaccine for any flavivirus, because the NIH group has proven you can do it for dengue 1, 2, 3, and 4. They didn't know why it worked until our work. Young has done it for Zika. We can probably do it for any virus. And Young is developing a new DNA launching methodology that could get the vaccine out extremely quickly. So can we do the same thing for other viruses? Can we find the Achilles heel of other viruses? And by the way, I don't mean to be chauvinistic about this. This is not the only Achilles heel for dengue. In fact, if you hit 2 of them at the same time, you make the virus too attenuated, so it's not such a good vaccine because it doesn't replicate as well. This virus replicates relatively well, but it doesn't cause pathogenesis. So this idea of getting generic vaccines is something we need to do. Will they be perfect? No. But in the face of a very, very difficult epidemic, they could be essentially critically important. A bit like what just happened possibly in Africa with Ebola. It's not a perfect vaccine by any chance, but it may have actually made a difference. So then, how else? Well, as I said, we know a lot of structural biology, a lot of molecular biology about the viral enzymes. We can develop small drugs that inhibit those enzymes. There's a lot of effort into that. But we've also proposed using small molecules and also biologics against host factors that are critical. And people worry because, well, you're affecting something in the host, but every time I take my blood pressure medicine, I'm inhibiting an enzyme, right? So there is a therapeutic index we have to worry about. But what we'd like to do is like a paper that, I'm sorry, I didn't put Danny Anderson's name here. Danny's a wonderful postdoc in Singapore. Together with Veronika Messling in Germany, we decided to take 3 very different negative stranded viruses from the Paramyxoviridae and Pneumoviridae, human respiratory syncytial virus, a very big problem. Mumps, not so much of a problem, but it is a problem around the world. And measles becoming a problem because of the anti-vaccine campaigns. And very distantly related viruses, and ask the question, can we find common host factors for all of them? This is one example of these massive RNA interference screens that we do, and we identify a series of host proteins, each of which is absolutely essential for the virus to replicate. Many of them are not going to be drug targets because you'll kill the human being, but many of them could be. So this type of analysis, and we repeated the same analysis with each of the 3 viruses, and identified approximately 300 factors that were essential for viral replication. Several of them, I would say between 20 and 30, absolutely essential. The virus essentially was dead without them. How many of those are druggable? We don't know, but we're pushing forward with this. And this is the type of information we would like to have for viruses, so that when a related virus comes in, we can make an educated guess that a certain factor will be a host factor. Finally, and this is a fun collaboration, very, very early, collaborating with Jean Yao at University of California, San Diego, and George Demopoulos, who's setting up the driver systems in Aedes aegypti. We're using these CRISPR-Cas9 enzyme that has been modified. Several groups have done this very pretty work that now cleaves RNA, and we're actually introducing them in Aedes aegypti in an inducible form. The guide RNA is constantly expressed. The enzyme is inducible. So it gets induced when they get infected with the flavivirus. And eventually we'll have a whole, like a CRISPR repeat of sgRNAs for every one of the flaviviruses. But we're starting with dengue too. And hopefully in the next year or so we'll have mosquitoes that at least in the lab will produce these RNAs, and hopefully they'll inhibit infection. Now there are many, many other groups around the world trying to do this sort of, using either RNA interference or CRISPR-Cas technology or expression of proteins that are antiviral. And then finally, we can't forget about our friends who are sometimes sentinels and tell us about diseases coming. And in this case, these are howler monkeys in Brazil, and they predicted the outbreak that happened in Brazil. The veterinarians and the people, the farmers, knew something was coming because the howlers were falling from the tree. So this guy, Jason Bobia, called me and interviewed me about this because it's a concern. If yellow fever spreads to the Caribbean, we have a huge problem, and we have a huge problem in Miami. So we better be aware of where monkeys are getting sick, because they told us by several weeks before what happened in Brazil. So we have to get veterinarians involved in this. So to finish this plea, I say that this really has to be an integrated from basic discoveries, and these are critically important. We actually want to base the whole thing on these basic discoveries. Then we have to transfer them to pharma, to medicine and veterinary medicine. And eventually, you guys are going to be the ones that are going to formulate these things and put them in the hands of patients. We absolutely need these last 2 groups here. It is a shame that people die of yellow fever in the world. And it's essentially a lack of will by governments and foundations. It's true, the vaccine is tough. It's an attenuated virus. You need to have it cold, blah, blah, blah. But, if we had the will, we could do it. We could vaccinate everybody that would be at any kind of risk. But governments are not willing to collaborate. Many of them are not until it comes close, They don't realize they have it. But that's for you guys for all of us to lobby. Okay, and to conclude, I want to thank the group in the lab. They're the people, they're the engine that drives this, and they're incredibly dedicated. In the lab in Galveston, this is my co-PI, Shelton Bradrick, who is terrific, and he and Ruben really led the whole Zika non-coding RNA work and collaborated with Pei Young for the vaccine work. And this is a group in Singapore. This is Julian, my co-PI. He's an expert in mosquitoes. Otherwise, I really wouldn't know how to dissect the salivary gland. So I'm very, very thankful for them. Very thankful to my amazing collaborator, En Young Oi, who is terrific, and for the very generous funding in across both sides of the Pacific Ocean. And I thank you for your attention. I'll be happy to take questions. I don't know if that's protocol or not, but I will. I guess it's not protocol. Okay, I'll ask myself a question. How long is it going to take to get that mosquito out? And I want to reiterate something that was said before. It's going to be a long time, mostly because of regulations and getting them out and making sure people are not concerned and making sure they're safe. Right? We don't want to do something stupid. But it's going to take a while. So stay tuned. Okay, thank you.